And hello, everyone, and thank you so much for joining us today on this live webinar with Kurt Swogger, the CEO of Biopact. We are very excited to be joined by Kurt. He is an incredibly experienced individual who comes to us to discuss his new company, Biopact, that he's been working on, which is a really, really interesting technology that we're excited to share with you. They're currently raising funds on Equifund. They've been rated a deal to watch by us here at King's Crowd. And this is an opportunity for you in the crowd to get to ask questions and learn more both about Kurt and the business. For those of you who don't know me, I am the founder and CEO of King's Crowd. King's Crowd is the first independent online private market rating and analytics platform. We essentially aggregate all of the deal flow across 50 plus platforms out there that enable you to invest in private companies and basically provide you vetting and due diligence services so that you can make informed investment decisions. So as part of that, we'd love to get on the phone with founders like Kurt to hear more about their story, what they're working on, how it's working, where they're headed, so that you can make a more informed decision. So with that, I would love to hop in. Kurt, thanks so much for joining us today to hear a little bit more about you.
Yes, sir. Welcome to this webinar, and my name's Kurt Swogger, as he said. I worked for Dow Chemical for many, many years as a chemical engineer in research and manufacturing. In fact, I can almost guarantee you've used my products. We created the materials that go into Crocs shoes or in plastic bags. And the way I got into this is we were looking to what can we do with nanomaterials in our polymers. And we had a spectacular failure in Dow. Couldn't get them to work. And so after I retired, a colleague and myself formed something called Molecular Rebar Design. And we decided we were going to make the nanomaterials work. And the real problem was, as made, there were bundles of millions of tubes and people couldn't get them apart. So when we did figure out how to do it, pretty obviously, and the individual tubes are nano-sized, in our case about 14 nanometers in diameter and about 1,000 nanometers long. And when you do that, you get some pretty extraordinary properties. And that's where we started. And to finish it, we were looking at what do these tubes do to people at the cellular level and the body level and so on. We were really looking for toxicological properties. And we discovered that they actually go into the cell without harming the cell. And in a nutshell, that's where we came from.
Got it. Very, very interesting. So to kind of break it down for those of us who aren't scientists, you've essentially created this very, very, very tiny, minute thing that could enter a cell safely and effectively. Is that correct?
Yes, they're about 10,000 times smaller than one of your hairs, and I can't visualize that. The only way we really can see them is using electron microscopes. But they're at the molecular level, at the size of about 5 times bigger than a rubber molecule or a protein molecule, which is why we call them molecular, and they act like in materials like rebar in concrete. So molecular rebar, and then what we found here is we use them a quite different way in the body. And like I said, we were looking for toxicological properties and discovered that we could go in a cell and we could carry pretty much anything we wanted to go into the cell.
Got it. So now I'm curious to hear, one of the things during our research of your organization is that, you know, there's an opportunity to take this technology and utilize it with cancer fighting drugs that are coming out. Tell me why the current standard of care, the things that are being created, can't safely and effectively enter the cell, and how it is that this solves that very problem.
Excellent. So pretty much all of us have heard about gene editing.
Sure.
And a fundamental issue is getting the editing tool into the individual cell so that it can adjust your genetic makeup. And currently, one of the major ways they do it for a treatment called CAR-T is to use viruses. So they custom engineer a virus, put on the agent, and then it goes into the cell and releases the agent to do your editing. Unfortunately, the virus kills up to 80% of the cells. It can also replicate later on and basically carry the virus into your body. And then it also doesn't allow you to do more than one edit at a time, practically speaking. So that's the current system. And the real problem is because you have to customize the virus, the treatment is $400,000 to $500,000 a patient, which is very expensive. It works really well 80% of the time and with only 3 injections and minimal side effects. It's just tremendously expensive.
Got it. So essentially we've created this gene editing technology that could be very powerful if it gets into the cell, but the problem is actually getting and delivering it into the cell effectively. And that's where you guys could come in and help solve for that.
That's correct. We don't pretend to know about how they exactly want to design their editing tools.
Sure.
If you want, our role is to get it in the cell without damaging the membrane wall. And then once it's inside, have the tool, I guess I'll call it, release from our tube. And then our tube just is very inert. And that's exactly what we do. The big advantage is we don't have to customize our tube. We might adjust the chemistry a bit on the surface, but it's very easy to do. You don't need 2 PhDs figuring out how to re-engineer a virus. With ours, basically you load it on and then put it into the cells, which by the way, never are in the body. Sure. They actually take and collect what they call T cells from your blood, return your blood, and those T cells you change. Once you have them changed and you grow new crops of the cells, they're all bioengineered to attack a cancer. You put them back in the body. So they're really customized medicine for you, the patient.
Got it. Makes a lot of sense. Now, from a, going to market, monetization strategy, all of those things, when we think about what you've created, curious if there are any actual gene editing tools on the market live being utilized by patients, and how do you get applied to those and make money off of that?
Yeah, there's 2 or 3 different, there's 2 companies with 2 or 3 different treatments. I think last year they did about 750 treatments, but again, call it $500,000. And by the way, we think they're losing money. There's maybe 50 other people trying to figure out how to do this. This technology doesn't just work with cancer, it works for other things, but the major focus right now is cancer. And our drive is how can we make it cheaper and better, and such that the cost comes way down. We don't have any viruses replicating themselves, so the quality is much better. And the time to take to get to cure is reduced dramatically. If you don't kill all the cells in the beginning, what they have to do is grow all those cells back plus, so you have enough cells to go into your body and replicate and grow so that you can attack the cancer. So if we start out with a huge edge, time-wise, it takes us a very short period of time to put it into the cell. We don't have to modify the vaccine. We don't have to wait for time for the cells to recover. So it's very quick.
Got it. Now, tell me about, FDA approvals. Are you looked at as a drug? Do you have to go through all these phase trials? Is it just being applied to the drug? How does that work?
Excellent question. So what we do is we never go back into the body. So we're literally a tool, or a reagent is what they call them in the drug business. So you harvest the T cells, bring it out of your body. We provide the tool that they can mount their gene editing. They basically culture it, let it grow back, and then all the daughter cells, none of them have carbon nanotubes in our MGMR in them, because our material doesn't replicate. So there's a way for us to separate the cells that still have the MGMR in them away from the daughter cells. And so there's a tremendous higher amount of daughter cells. So our tubes are never really put back into the body. Because of that, we are not as the individual tubes regulated. The chemistry is still regulated that our CAR T makers deal with, but we are just a reagent, and we like to keep it that way. All we have to use is good laboratory practices to make our material.
Got it. Excellent. So basically that whole approval process becomes much more simplified and easier to go through. So can you tell me a bit more about where you guys are in terms of development?
Yes, we've demonstrated it very nicely in our lab. So we actually get cells, and in this case T cells, and we've shown that we can load on various proteins, whatever, talons is what they call them in CAR T, and that we can go into the cell without damaging the cell, and it does unload. Now, we don't claim to be the guys who figure out what to edit. That's other people's job. Our job is just get it in the cell. So we've assigned a research agreement, I guess we'd call it, with one of the larger players, and they are in the process of validating all the things we've said. And we've got lots of data, but of course, a CAR T maker wants to do it with his system, and we thought they were just going to look at one system, and it must have got excited because they have 4 or 5 they want to try out. So our job is they send us chemicals, we'll put them on the tubes, ship them back, and then they do the editing, if you want.
Now, in these research agreements, curious to hear from you, what IP do you guys own? How do you protect what it is that you've designed? If you're having these large firms work with you, these large pharmaceutical firms, how do you make sure that they don't just come in and do the same thing or, replicate what it is that you're building?
Yeah, that's where the Dow experience helps out. We were fortunately involved in a huge patent effort, so we've got a lot of that same team in place. So we actually have the composition of matter, the individual tubes, and how you coat them and what you put on them. We have covered by patents right now, 78 patents, I think it is. And so what we can do is block anybody from doing what we do. Big companies are as trustworthy as your patent position on copying you. And so we can block them. We, of course, have all kinds of agreements, but owning the patents is really essential.
Got it. So you own these 78 patents, you're going and you're working with these larger pharmaceutical firms. So that's great on the cancer front. And I know that's where a lot of the focus is. Can you tell us about what some of the other applications look like where this could be applied?
Well, we've heard of applications people are doing it. By the way, it's done virtually the same way. They harvest different kinds of cells. So I think a lot of immune diseases are fair game. Actually, even muscle repair or nerve repair, which we're not quite sure how they're doing that, but that's what they're doing. And maybe perhaps helping, diabetes. Pretty much any type of infection where your body really isn't fighting it, and you can bioengineer your cells to fight it off. So you're making your own drug, if you want, internally.
Got it. So it sounds as though you're kind of playing to this trend line of bioengineering and gene editing. So just to take a step back for a minute here, can you tell us more about that trend that you see and how you see this playing out? I know that there's been tons of talk and tons of excitement around gene editing, but then I would say movement versus how much talk there's been has obviously been slower. So how do you see this playing out over the next, 3 to 5 years?
Well, I think the trend is moving from treating your body to treating your cell. So they're calling it the cellular revolution. And so lots of different ideas, concepts to treat your cell so that it treats you. All we want to be is the mechanism of choice to get it in the cell, because every one of those has to go inside that cell. And if we stay outside the body, at least with this particular piece of technology, we again avoid FDA. Our partners will have to deal with it, but we can just go in and be a chemical, if you want.
Got it. From a monetization standpoint, I know we started to hit on it before, but when you work with these firms, how are you compensated for providing this technology?
Actually, pretty much like a drug or pharmaceutical. We have milestone payments, and that's when we sign a license. We get payments when they've tested on a few patients and it seems to be working. We get patients when they start going through their phases or the clinicals. And then we get paid once they commercialize it. And then, of course, we get to charge them for supplying the material. So it's pretty much like any other pharmaceutical. And so what we'll end up with is a longer term as a royalty, but the milestone payments are pretty hefty.
Excellent. So your team today, what does it look like? What are they working on? And who do you need to add to the team to kind of take it to the next level as you do begin to commercialize?
Good question. So the parent technology company, Molecular Rebar, is, if you want backup, we'll supply materials. If they need a slightly different tube, we can do that. The people who are really working on making it work in this application, there's 3 or 4 very young biochemists and biophysicists. I never knew there was such a thing as a biophysicist, but there really is, and they study what goes on inside that cell, which is pretty neat. And so what they do is they worry about how do we get whatever material somebody wants on the surface of the tube, make sure it goes into the cell we're aiming for, and then make sure it releases inside the cell. And there's a bit of technique to make that happen. And that's what that team does. And what we need to do is add 1 or 2 more of those kind of folks, but then we need to add some people that will call on customers, go out and spread the word if you want. And what we want to do is continue to add more and more customers, or they're really pharmaceutical companies, to our list. So we don't have to worry about going and talking to consumers, but we do have to talk to the people that are going to make these drugs, whether it's CAR T or CRISPR or for cancer or for whatever. And so we really need to add, they call them business development people.
Yep.
And then I'd like to add a CEO that is familiar with the drug industry. I'm very good at chemical industry, but you need to build up contacts there.
Makes sense. And I could imagine it's a slow and challenging process dealing with these very vast, large pharmaceutical companies. When you think about timeline of getting up and running with some more of these organizations, what does that look like? Is this a 12 to 15 month type of process?
Well, what we've been finding is we go out and talk in conferences, and we talk, and we end up usually getting one group for each one, about 1 every 3 months. And we think there's some other ways to speed that up, but if we get too many, we can get overwhelmed. But we certainly can absorb some more, and we'd like to get 4 or 5 of them. And we think we have the winning technology. They're going to beat out the other guys, and we're aiming to reduce the cost at least in half. And obviously, if you can sell something for $200,000 versus $400,000, we know how that's going to play out. So we need to get a few of those going, and that's what we're in the process of doing. The only tricky part, of course, they don't want us to talk about their competitive edge, so that makes it more interesting, right?
How have you been funded to date? And it does seem like it's a heavy lift to create this type of technology and have these biochemists on board. So what is the funding requirements you think you need moving forward?
Well, to date, we have 2 partners that really own this technology, our group and another. And so we've been putting the money in and supplying the technology. But now that we're getting closer to market, we think it's time to help spread our risk and bring in more cash to accelerate it. Now that we've proven it out, we have the patents, it's time to go make money.
Excellent. So just before I continue with questions, I did want to let everyone know listening in that you could enter your questions into the Q&A, and over the last 10 minutes, I'd be happy to try and get to them. So talk to me a bit about what exit strategy looks like for an organization like this. You have this technology where you're essentially licensing it to various pharmaceutical companies. Is this a buyout by a large pharma company, or is this, something where another chemicals provider is coming in and wanting to own this asset to be in the pharma industry?
Well, I don't want to sound facetious, but whoever puts up the money, we're perfectly happy to go with. It makes sense for a chemical provider, or they call them reagent, to pick this up, because then you can sell to many pharmaceutical companies. Or a pharmaceutical company might want to come in and say, hey, we want an exclusive in this particular area. So those are both exit strategies. Or some of these pharmaceutical companies are getting so big that they have a whole range of things they could use. So we're looking at either one. Right now, from my point of view, we need to get it so somebody's saying, yes, it works, in actually putting it in patients. And the nice thing is, because we're a reagent, we can go in and replace what they're doing already, and they don't have to go through the whole FDA process. And so we're working with people that are in that situation. That makes it much faster. But the exit strategy after 2-3 years is to definitely be purchased.
Got it. What does a 12 to 18 month product roadmap look like?
Well, as we said, we're working with a player, not to be named, who's already in that game. I would be very disappointed and surprised if we weren't in his queue pretty quickly, simply because we're so neutral. We're just a reagent off to the side, so we don't actually ever want to get into body. What they have to do is, and they already are there, have a drug registered, or I guess they don't call them drug now, but tool registered.
Makes sense. When you think about challenges that you guys face moving forward, what's the thing that concerns you, worries you? What people always say to the founder is, what keeps you up at night as you think about progressing this business forward?
Actually, I think we've hit on a little bit is now get the word out. I mean, we've done it enough with different materials, being transferring things inside the membrane. Besides CAR T, we put up many other things in. So we know the technology will work. And so far, we haven't found anything that even looks like a showstopper. And particularly where we keep everything outside the body, we're like gold miners. The people who really made the money weren't the gold miners, they were the shovel providers. And we want to be everybody's shovel, if you want. So how do we get that word out there? How do we penetrate, you alluded to it, the maze, which is some of these larger companies. They protect all their scientists from people because they want to make sure they stay focused. Well, we want them to get excited. And the way to do that is continue to go to conferences. We publish papers. I think they are on our website. How do we get the word out, bring some people in that know how to do that?
Got it. So a question that just came in from the crowd is, nanotechnologies have existed for a long time. What has prevented someone else from creating a technology like this, and what's preventing them from doing it in the future?
Excellent question. And I'll stick to the one I know the best. Actually, it didn't exist. They talked about nanotubes, but they never had nanotubes. They kind of look like a cotton ball. And that cotton ball is 2 to 10 microns, which makes it 5 times smaller than one of your hairs, and about 1,000 times bigger than a nanoparticle, or 10,000 times. So they have these balls of stuff, and what really is a big market, it's not cotton ball, it's the thread. And what we have is a thread, which is pretty unique, and particularly the long skinny thread, which is what we're talking about. And what we have is a very good patent portfolio. Composition of matter, it doesn't matter how you do it. If you have the skinny threads, you're going to have to pay us if you want. So a patent portfolio, getting a position in the marketplace are very essential. And so it was really a misnomer when they talked about carbon nanotubes. Never had them. They had fuzzy cotton balls, which aren't the same thing.
Excellent. I appreciate that. Another question that came in from the crowd, I'll read it off from them is, I would like to know what patents and any other IP, et cetera, are actually owned by the entity that is raising money, Biopact CT. Does Biopact actually own anything other than the licensing rights? If so, what? And what guarantees does an investor have that Biopact Ventures and Molecular Rebar Design LLC don't license the tech with other entities?
Excellent question. Because of the complexity of the patent situation, MRD owns all the patents. It has assigned rights, exclusive rights to ventures, which in turn assign certain rights to Biopact CT. And so it's all done by contract. I guess if MRD tried to, and MRD owns half of it, so it's in our best interest. But even if we didn't, and we tried to license to another entity, somebody'd bring a suit and rightly win that we're licensing the same thing twice. I don't know what it's exactly called in the patent word, I call it fraud. But we have the exclusive license for medical sits and ventures, and for, I'll call it CAR-T, and the intracellular stuff belongs to Biopact CT.
Excellent. And one last question from the crowd is, they have a family member that has cancer, where they're up for a gene editing solution. How would this improve their experience of going through these treatments from what they're currently getting today?
Well, first thing, it would cost half as much, so they got a shot at getting it done. Second thing is there would be no possibility of a virus or fragment of virus in the cell. The trouble is, once you get one in your cells, it keeps replicating it. And so we take that out of play. And the third thing is, if your biochemist wanted to do more than one edit, it's very difficult to do with the virus, because basically, if you go through 2 treatments, you kill almost all the cells. Our material, you can very nicely do sequential editing. Now, so practically speaking, we give a lot more flexibility. So we consider it cheaper, safer, better.
Cheaper, safer, better. Always a good thing. So for those who are on the call, who aren't on the call, that are considering investing in Biopact and they're kind of still on the line and they're not sure, especially because of the risks associated with making kind of a biotech investment, what's kind of your final pitch to them as to why they should think and consider making this investment?
Well, I think, first of all, good news is we're not a pharmaceutical. We're, as I said, the shovel, not the gold miner.
Right.
And so the risk is tremendously lower. We will be much quicker to market because, again, we're a reagent. And I think that the CAR-T is a marvelous chemistry if we could just get the cost down. And I think we will be able to enable people to do that. And instead of in 10 years, 7,500 people getting the treatment, we ought to be trying for half the population. And I'm very interested in that. As I mentioned in our clip, my brother-in-law, Tony, is a real life person, and he's going through the second treatment right now. And what we want to do is have this thing ready for him in about 2 years, which is when he'll need his third treatment. So I have a very personal interest in it.
Yeah, no, absolutely. Well, from our perspective here at King's Crowd, there's always 5 things that we look at. It's market size. And from a market size standpoint, you're going into really a newly emerging market, right? But an obviously very fast growing and large market opportunity. We look at product and service differentiators. So from a product standpoint, you're creating a technology that, frankly, there's just not really a good solution today. And you're creating an answer to that, which is a really, really big deal. Taking it from being a virus-based thing, which could actually infect and make a patient sick and kill their cells and all of these terrible things, you're creating a solution that can prevent all of that, which is incredibly important. In terms of the deal, I think from the standpoint of the technology that you're creating, how close you are actually to be able to go to market, the fact that you're mitigating a lot of the issues with FDA approvals is a really big deal. And so the terms from that perspective, if you look at biotech deals out there, and I know you're more of a shovel than biotech per se, but I think it plays in the realm and it's well within reason. The business model, I mean, end of the day, that is a challenge in being in this space, but you're playing with the same business model that everyone else is. And I think that's totally fair and reasonable. And then founder experience. You've been in the chemicals industry, you've been around the space, you understand and recognize where you need to fill in the gaps, which I think is really, really important. And so from a whole perspective, we get really excited by the opportunity. Obviously, again, there's inherent risks in investing in something like this. I think anything that plays even in the realm of biotech does become incredibly risky, just because dealing with humans and how it interacts with humans and going through that whole process, we just never fully know until we're down the road a bit. But at the end of the day, I think there's going to be plenty of acquisition opportunities for a business like this. But I really appreciate you taking the time today, answering all of our questions and helping us understand this investment opportunity better. It's been a pleasure chatting with you, Kurt. Thank you.
Yeah, thank you so much for the time, and thanks to everybody who took the time to listen to this. It's a pretty exciting area that I never dreamed I would ever get into. And
that's outstanding. So for those who are on this, you could go to equifundcfp.com and you can place your investment there if you're interested, and you could also read up more on the business. So thanks so much and have a wonderful day, everyone. Take care.
Bye-bye.